Hi everyone, my name is Ria Nabar, and my project is a comprehensive review and evaluation of the diagnostic methods of multiple system atrophy. And before I go into what that really means, I want to talk about my motivations for this project. So this is a picture of my grandfather and me, and my grandfather was diagnosed with multiple system atrophy. And as a child, I kind of witnessed the symptoms build up over the years. I saw how he was less active and he couldn't move around as much as he used to. And after his death, I found out that he was actually originally diagnosed with Parkinson's disease. What this did is it prolonged his disease management and it made the true disease, multiple system atrophy, progress even further. So, what is multiple system atrophy? It's a rare neurodegenerative disorder, and it has no known cause or treatment. This is because scientists and researchers don't know the underlying mechanisms behind multiple system atrophy, and it makes it a really difficult disease to identify. It is classified as a synucleinopathy, and that means that it has abnormal amounts of a alpha-synuclein protein. This is a protein that is really fundamental to multiple system atrophy, and I'll delve into a little bit more about its significance in the later slides. The main thing I want to emphasize is the overlapping symptoms that multiple system atrophy has with Parkinson's disease. They're both neurodegenerative disorders, and these overlapping symptoms make the diagnosis really, really difficult. So the main issue here for the diagnosis of MSA or multiple system atrophy is that misdiagnosis. And what this causes is that very late diagnosis and not effective disease management. So my objective was to evaluate methods that counter misdiagnosis, as studies have primarily focused on only identifying disease presence. So the studies say, hey, okay, we know we have this disease, but we don't really know which one. So we have a need for differentiating more between diseases and distinguishing between them. Um, okay, it's not really letting me go to the next slide. Okay, there we go. So a little bit of background information is that there are two types of multiple system atrophy, cerebellar and Parkinsonian. The cerebellar one has a lack of coordination associated with the cerebellum, and the Parkinsonian one is associated with motor abnormalities such as tremors and stiffness. Some of the symptoms are autonomic failure, cerebellar dysfunction, and Parkinsonism. And what I really want to point out here is that a lot of the symptoms and types are actually named after Parkinson's disease, just emphasizing how much the disorders are so similar. Some of the affected brain regions are the cerebellum, the middle cerebellar peduncle, and the putamen. These are all associated with movement and motor learning and coordination. And the brainstem, the parts of the brainstem that are targeted by multiple system atrophy are the pons and medulla, which regulate autonomic functions such as heart rate and breathing. So the diagnostic methods I'm going to be talking about are adjusting the diagnostic criteria and biomarkers. Diagnostic criteria are a set of symptoms and tests that are used to identify a certain disease. These are basically guidelines that say, hey, we have this certain disease, and they're really crucial to having a correct diagnosis. The Movement Disorder Society outlines 4 levels of disease progression within the diagnostic criteria for MSA, and these are based on disease severity and severity of symptoms. So from possible prodromal, which is the very early stages of MSA, to neuropathologically established, which is actually an after-death or post-mortem level. So that helps scientists establish how the disease has progressed over time and what that means. The stages really emphasize the atrophy, level of atrophy or neurodegeneration within the brain structures. And on the next slide, you can see what this really means. So for A, on the very left is an MSA or multiple system atrophy brain. In the middle is a Parkinson's brain. And on the very right is a healthy brain. So I drew these illustrations from MRI images and to really emphasize that the structure of the brain has deteriorated. So as you can see from the healthy brain to the MSA brain, you can see how the cerebellum, which is the blue structure, has deteriorated through the diseases, and the red lines indicate where that cerebellum would be if it were a healthy brain. On the next slide is a similar view. It is more of the middle cerebellar peduncle, and the letter A is multiple system atrophy, B is Parkinson's, and C is the cerebellum. And you can see just how much that cerebellum has deteriorated in multiple system atrophy versus even Parkinson's and the healthy brain. And here you can really, really see how much that structure has deteriorated and how it has affected the brain. So, a little bit of the pros and cons for the diagnostic criteria that we have now. The pros are that it's really, really great for identifying the presence of a neurological disease. They're great guidelines for saying, hey, we have these symptoms, we have this condition, and we know that we have this disease that's present. And if confirmed, these MSA levels help establish that timeline for disease progression, allowing for correct disease management. However, the issue is with actually confirming that we have a multiple system atrophy disease. The lack of criteria that distinguish MSA from other disorders is really evident, and this contributes to a misdiagnosis. We need more criteria that say we have this differentiation between multiple system atrophy and Parkinson's, and because we have this specific symptom, it means is one and not the other. And right now, we just don't have that. The next method I want to talk about is biomarkers. Alpha-synuclein, as I mentioned previously, is a really fundamental biomarker in MSA. It regulates neurotransmitter disease, including dopamine, which is crucial to movement, and it accumulates through neurodegeneration. Basically, it clumps up together through a process of misfolding, and this reduces its function and makes it work in a way that it shouldn't. And so a method called protein misfolding cyclic amplification amplifies this misfolding process and makes it easier for researchers to see the aggregation in MSA versus PD patients. So a study found that MSA patients had significantly higher amounts, saying that the aggregation was worse and therefore the symptoms were worse for MSA versus PD patients. What we need is for more studies to replicate this finding so we can have a dependable threshold. Once we establish that we have this number of aggregation, then we can categorize, okay, this patient has this level of aggregation, so they're categorized in MSA versus PD. Another method is protein magnetic resonance imaging, which measures metabolite concentrations. Two metabolites that were measured in a study is N-acetyl-aspartate, which measures the health of neurons, and myo-inositol, which is an indicator of how the nervous system responds to injury. So a study measured the pons and medulla of 12 MSAC patients or cerebellar patients, and it found that there were significantly higher levels of myo-inositol in MSAC patients, and this is compared to healthy controls. This indicates that there was greater injury because that nervous system is responding that much more, and lower levels of an acetyl aspartate. So this is decreased neuron health and indicates that this makes sense because there is neurodegeneration. For future direction for this study, I would want to investigate metabolites in the cerebellum because it is such a prominent structure in MSA, and seeing how those metabolite concentrations would differ if it were measured in a different brain structure, as well as including MSAP, Parkinsonian, or Parkinson's disease patients, because if we saw any difference in metabolite concentrations, it would be a really great indicator as to distinguishing between the diseases. And now to wrap it all up, just the key in differentiating between MSA and other diseases is in that disease severity. So as we saw there, people with multiple system atrophy had more severe symptoms in aggregation, in atrophy of the brain structures, and metabolite concentrations. So those are really key. And if we can replicate those symptoms in consistent manners and across studies, we can categorize them effectively in order to distinguish between diseases. And the reason this is also important is because multiple system atrophy and Parkinson's disease progress very differently. MSA progresses faster, and individuals with MSA have a lower life expectancy. Because if the symptoms were the same and they were treated the same, it wouldn't really matter which diagnosis you got. But however, since there is different symptoms management, for example, people with Parkinson's often respond to a treatment called levodopa. However, people with multiple system atrophy do not. So that's different symptom management is really one of the main reasons that we want to have that distinguishability. And so, effectively managing this disease progression requires that accurate and distinct diagnosis. And this really is only possible through improving methods and resources that we have available today. Thank you.
Thank you, Ria. Um, Question for you. I mean, it was a very well presented research, and you seem to have gained a lot of knowledge. I'm curious because you mentioned it was a personal experience with your grandfather that kind of took you down this road. Is this also, though, a field you plan to pursue as you continue in your education, or was this kind of a one-off because you wanted to learn more about your grandfather's condition?
It was a little bit of both. So when I came into Polygence, I knew that I kind of wanted to do something with neuroscience, and I knew I wanted to pursue research because I just found that contributing to like more developments in the world was really interesting to me. So I wanted to pursue that. And kind of when I learned more about my grandfather's condition, I decided, wow, this really isn't researched at all because it's such a rare condition. And I wanted to learn more about that. So it was kind of a little bit of both.
Yeah, and it's a really quite a conundrum, right? Because they have similar symptoms as Parkinson's. It's a rare condition, and it
Mhm.
does seem like most of the symptoms that you would be able to classify as MSA don't actually happen until they'll later down the line where they might have been already misdiagnosed for
Exactly.
Parkinson's, right?
Yeah.
I mean, what a troublesome situation, but I appreciated the research. So, of the things that I thought were interesting, you mentioned like the four the four levels of how they kind of do the diagnoses, but in your own, I was even going to ask you the question, but you already answered it, which was that even with that we still have a problem with misdiagnosis because those are also similar to how you would test Parkinson's.
Yes, exactly. Yeah.
Right. So what about the I did find the biomarkers stuff pretty interesting. So you had mentioned, for example, that people with MSA actually have more severe symptoms. And in your research, did you find out how soon that occurs so that doctors can start to maybe classify it as MSA, or is it a little bit too far down the line at that point?
I think most of what I found it was a little bit too far down the line, and a lot of the studies were actually post-mortem. So it was kind of like, okay, we found this aggregation and these different levels of metabolite concentrations a little bit too late, and it offers a good guideline as to what we're working towards, but seeing how we can improve upon that is really important.
Yeah, it seemed also really dangerous because didn't you mention in your research that sometimes if you misdiagnose it as Parkinson's, it actually accelerates the symptoms of MSA, correct?
Yeah, yeah.
Wow, that's wild. Well, I really enjoyed it. That was informative. I had not seen anything like this before, so I really appreciate it. Thanks, Ria.